Good afternoon, thanks. I'm Tony Oro. I'm one of the co-directors for the Maternal and Child Health Research Institute here at Stanford, and wanted to welcome you to our MCHRI seminar series. This is our monthly series, now virtual, which allows us to broadcast important, new, and cutting-edge research that's being done here at Stanford to impact maternal and child health. And today we have saved the best for last for this this academic year, which is Mike Snyder. And so next slide. What we wanted us to do is to highlight the beginning seminar for next year, which is September 11th. So put that on your calendar on Monday, September 11th, which is the first Monday of the year, which is novel strategies to prevent gender-based violence among adolescents. We have an amazing group of folks here. And then next slide. Also wanted to point out the rest of the series, that the next couple in the series, which includes climate change and vector-borne diseases in October, our MCHRI seminar in November, and then the new professor of population biology, Melissa Bondi, will talk about pediatric, adolescents, and adult brain cancer. Really a great seminar, so put that on your list. And then next slide. Our MCHRI virtual symposium is November 6th, which will be keynoted by David Williams here, who's the Senior Vice President and CSO of Boston Children's Hospital, talking about pediatric cell and gene therapy. And so this is really a wonderful and actually an innovative interface for you to display your research. So please put it on your radar, and we'll have a call for abstracts coming up in the next couple months. But today I wanted to introduce, next slide, our education series symposium committee. And the next slide. I just wanted to say for today's symposium that we have a questions and answers function, so please submit them. The chat function is disabled, but please submit your questions for Mike's seminar today via the question and answer function, which I'll then quiz him at the end. So the more questions, the better. So please put them on the Q&A symposium. And then we're going to send you an online survey with your feedback for today's talk. Next slide. So today, we're so fortunate to have Mike Snyder, who's the Stanford Ashman Professor and Chair of the Department of Genetics at Stanford, and then actually Director of the Center for Genomics and Personalized Medicine. I don't think he needs a big introduction, except the fact that he really has been a very important part of the MCHRI faculty family, really providing the omics in maternal and child health research. He's was actually a postdoc here at Stanford, went to Yale, came back, and really is a really important leader in functional genomics, which he'll tell us about today. I think importantly, understanding how we can then really connect omics and big data with individual health in terms of the pediatric maternal and child health individual health record is really the connection we're trying to make here in terms of bioinformatics and also measurement data. And so, Mike, thanks so much for joining us today. I look forward to your talk.
Great. Well, thanks, Tony. Thanks for having me, and it's really great to be here. Here we go. Chat. All right, well, as Tony says, I'm going to tell you about our work using big data in health, and some of it, for those who've heard me speak before, you may have heard the beginning part, but I've got a lot of new stuff at the end for those of you who've heard me before. So I'm going to start with a premise that's one of my favorite, which kind of offends a lot of people, but it's I believe the way we do medicine has a lot to be improved in many different ways. I think many of you will appreciate we tend to be very focused on illness. As such, we're very reactive. We typically measure very few things when people are healthy. And I would argue even some of those things, you know, maybe not, they're not the best measurements necessarily. When you're healthy, you're typically measured very infrequently. And perhaps the most important thing here is that nearly every decision about your health is based on population-based measurements. And yeah, I think we should all be very focused on keeping people healthy. I think most people would agree with that. As such, we should be very proactive We're certainly capable of measuring many, many more things than we currently do. The frequency, I will argue, should depend on what your risk is and your personal trajectories for particular health biomarkers. And that, I think, is all derivable. And perhaps most important, again, we should be very individual based. And I know people will say we are, but I don't think it's at the level it could certainly be. And then, of course, this is a major theme of the medical school. We should be very focused on precision health. And so much as just, one suit doesn't fit all people, we really do need to customize health and medicine management in an individual way. And to illustrate this point further, this is actually a measurement of healthy people's oral temperature. So we're all taught that people's oral temperature is 98.6 when they're healthy. And it turns out, first of all, that median value is not accurate for adults anyway. And this is one of several studies that all put it a little bit lower, 97.5. But the more important point is that there's a spread. So this is the 25th quartile. Again, this is healthy people measuring their oral temperature. 94.6 is the 25th quartile, 99.1. And so in today's world, if this is your normal baseline temperature at 94.6 and you come in 4 degrees higher than you should, meaning you come in at 98.6, people tell you you're healthy, everything's great, what are you doing here, go home, when in fact you're up 4 degrees Fahrenheit, and I guarantee you're not healthy if your baseline is that lower number. By the way, mine is never 98.6 unless I'm unhealthy. Mine's 97.3 and seems to have dropped a little bit over the last 8 years. So the point is you do need to know your baseline. It's a theme I'll bring up multiple times through today's talk. So your health, obviously, it's influenced by many things. The genome used to get a lot of attention, but we know all these different things will contribute to your health. And we know that in a very qualitative fashion. And I would argue we're in a world where it's now possible to quantify a lot of this information. Some of it easy. You can actually measure your exercise pretty quantitatively these days in a pretty easy fashion. Food's harder, but still doable. Stress or measurements for that, and then various pathogens and exposures. These can all be measured in some fashion, but certainly their effects can be measured on you too through molecular measurements, through your blood and urine and other measurements to be able to get some indication of health state. And so I would argue we really want to understand this in quantitative terms, because I'm a believer that people will be born with their genome sequenced in the not so distant future, and then you'd like to understand what kinds of exposures put you at highest risk for certain health outcomes. And so that's where certainly I'd like to see things head in Mike Snyder's world. As I said, we can now measure the effects of these things on people by doing all kinds of measurements. And this is a project we started now a little over 10 years ago, I guess 10 and a quarter. We set up something we call Personalomics Profiling, where the idea is we would measure the genome once, and in its most deluxe fashion, we'll measure 14 different ohms, mostly out of blood, a little bit out of urine for some of them, and then microbiome we get actually out of gut and nasal, although we've just gotten data back on tongue and skin, so we'll know more. And again, most of these come out of blood, so your epigenome, transcriptome, proteome we'll measure out of PBMCs, but we'll also measure plasma, the proteome, cytokine from the immune monitoring core. We've done a lot of work over the last several years to improve metabolomics and lipidomics and profile these very, very deeply. And actually with some new technology, we think are quite powerful. And then about 7 years ago, we added on wearables and smartwatches and other sorts of things. We do deep clinical tests, questionnaires, stress, etc. We also, for a subset of individuals, will run stress echoes and various glucose control measurements. OGTT everybody gets, but others are more selective. And so again, we're doing these deep profiles. And so one aspect is the big data, where we're collecting very deep molecular and physiological data. But the other aspect is that it's longitudinal, meaning we'll sample people when they're healthy roughly every 3 months. And then when an adverse event comes along, like a viral infection, very relevant to today's world, we'll sample many more times, typically 5 to 7 additional times. And the goal here, out of all this, and oh, by the way, we're doing this on 109 people, and we've been doing it roughly 7.5 years for most people, some a little longer, some a little less. Me, 10 years, I'm one of the participants. And again, why are we doing all this? We're trying to understand what it means to be healthy. These are generally sampled when people are healthy. How do they differ? How do people differ from one another when they're healthy? How does that change over time? And then most importantly, what happens at earliest signs of disease, and can we use these advanced technologies to be able to better manage people's health? Now, these all are adults. It's an older population of 53.4. Amazingly, this crowd in particular may appreciate it. Seems very hard to run studies on kids without the IRB kind of like going all over top of you. But anyway, I think we need these same kinds of studies on kids. I'll just say we've launched, which I won't talk about much today, but we are doing, we want to do a metabolic profile on every kid that walks into the clinics at Lucille, at Packard, and also every kid that's born. That's actually happening. We're launching that, and that's been running quite well. Separate topic though, I won't talk about today. So anyway, we're doing these very, very deep profiles. And so to get at the last question I raised, can these advanced technologies be used to better manage people's health? The answer turns out to be yes. So just from the first 3+ years of data, a little over 3 years of data, we had 49 major health discoveries spanning a wide range of areas. And if you count hypertension, it's 67. And so again, they'll cover things like heme-oncology. We call it some of the early lymphoma. That was through imaging and biomarkers. I'll show you examples in a minute. 2 pre-cancers, MGUS, smoldering myeloma. 2 people with serious heart issues, one picked up through genetics, another from a wearable. Some people with diabetes who didn't know it, and so on and so forth. So no, the key out of all this is that no one technology found all these things. We actually needed the combination of molecular profiles, genomics, and also some level of imaging where it was applied to follow many of these things. Even wearables found some important things. And I'll say a little bit more about that later. But we can, so together they found these different sorts of things. And so again, just to run you through this, the genomics did find a fair amount. There were 12 people, actually 13 with clinically actionable information. One's a pharmacogenomic variant. 12 individuals have mutations they should know about that's important for their health, like BRCA mutations. I'm sure this crowd's familiar with that puts you at high frequency high risk for breast or ovarian cancer if you're a woman, and some of these others. So 3 of them actually had very, very actionable information in the sense that one person had a mutation, this cardiomyopathy gene, and turns out fairly young guy actually has a heart defect. And that was actually caught. Now he did a follow-up stress echo, and he's actually on medication. Another individual, also fairly young, had a mutation here, puts him at high risk for certain cancers. Turns out they wound up getting a whole body MRI follow-up, and they had early thyroid cancer, which is one of the risks. And that was removed, and they still have most of their thyroid, so they're not on replacement therapy. And then here's another one down here. This individual actually there are 9 diabetics in the study. I forgot to say, we thought we were predisposing it towards diabetes and prediabetes. In the end, it's very similar to national population. 9 individuals were diabetic coming in. They're all thought to be type 2, but this individual turns out to be a MODY mutation, a very different kind of diabetes, and it's probably been on the suboptimal medication for years. This is pretty valuable. As I say, there was another person learned something important from they had a heart attack, and they were on a drug that predisposed them to another high frequency heart attack. So we got them off that. So anyway, those are, genomics did find a lot. Imaging found things. So we found one person with early lymphoma. Like I said, 2, and other biomarkers suggested that as well. 2 people with pre-cancers. This one individual has also fairly young, had way too much IgM compared to everyone else. So there are 1,000 samples here. This individual had given 2 at the time. You can see just outlier analysis, very different from the rest of the pack, and follow-up said, yeah, way too many IgM cells. And so they'll be, if you will, followed closely for this, because 15% of the time it can convert to aggressive cancer. And smoldering myeloma is another situation very similar. So again, these different markers would uncover different things, and the key aspects were that we were taking multiple measurements and we're following people over time as well in some cases. So that really helped. 6 people with plaques in their arteries. Of one of the 6, they're not huge plaques, they're modest plaques, but the recommendation is increase your statins. And in my case, it actually does seem to be making the plaque go away. So again, this is actionable information. Entering the study, we're doing quite a bit in the area of cardiovascular disease, thanks to our collaboration with Francois Haddad, and also diabetes here. We work with Trace McLaughlin on that. And 9 individuals were thought to be diabetic coming into the study. It turns out when we actually measured them, 2 were diabetic who didn't know it, and a lot were pre-diabetic and didn't know it. 9 out of 10 pre-diabetics don't know it. You may know that. I guarantee a lot of you listening to this seminar probably are pre-diabetic, spiking your glucose away in unhealthy fashion. And I would suggest you might go get a continuous glucose monitor. I won't have time to talk about that today. It's really fascinating. But anyway, one of the things that was of high interest to us is how do people become type 2 diabetic? And so because we're profiling people over time, what we discovered is quite a few had diabetic range values, and 9 were clinically diagnosed as type 2 diabetic, and others became pre-diabetic. And so the question is, how do people become diabetic? And the answer is 2 people are very classical. They gained weight, their microbiome diversity dropped, and they, quite frankly, hit diabetic range. And yeah, and then, but there are 7 others who didn't gain weight. So how did they become diabetic? Well, it turns out 5 of them gradually became diabetic, and here are 2 examples here. So this orange line is actually fasting glucose, and gray is actually hemoglobin A1C. And what you'll notice is this individual actually got there through oral glucose tolerance tests, actually gradually went up from a pretty high value to into diabetic range. This individual got there first through, I think fasting glucose was first diagnosed, but hemoglobin A1C was not far behind. So, and then they actually got treated, so they started going down. So these folks basically gradually got there. But what was interesting is there are 2 individuals, one of them's me, this is me in fact, there's about 8 years of data. And I spiked my way into diabetes as though something triggered it. So initially I was marching along fine, and then I shot through the roof. I actually went to get measured for an insulin resistance test, and I was actually abnormally high, and we were a little surprised, but it actually went up even after that test. It came up at about 3 weeks after a nasty respiratory syncytial virus infection. And actually, as we were measuring me right after that, it's pretty clear. Well, basically, I hit diabetic range. I had a pretty bad lifestyle in the sense that I ate lots of sweets and had been biking some, but I got it under control initially by cutting out all exogenous sugars, increasing my biking, doubling, and it started running, and I brought it under control. In fact, it was so boringly low, I stopped looking at it. The point out of all this is that we think the viral infection may have triggered it, where it's associated stress response. Anyway, it was so boringly low that I stopped looking at it, did the perfect control. Someone else looked at it when I was here and said, Mike, you realize your hemoglobin A1C is back to 7.1, 7.0 actually at the time. So no, I had no idea. Sampled me a few times. We looked back, and it turns out I had stopped running, and guess when that was? Right there. But I also had a viral infection, so it's not 100% clear whether it's the running, the viral infection, what triggered it. But either way, I started running. I brought it down lower, but I could never get it down to baseline levels again. And it gradually kept creeping up. Remember, this is 8 years of data. There's a lot of data points here. I've given 300 samples during this time. So what happened was it kept creeping up, gradually creeping up. And when it got to about 6.4, which just bounced a little bit there, but it was running right around the threshold. So I said, all right, it's going up and not really getting it under control. I'm running about as much as I can, 4 days a week, 4 to 5 miles a run. So I said, and I've been reading with Latha about, glucose homeostasis being better managed by muscle mass. So I stopped running. I switched to weightlifting. So you can't tell, but I've been weightlifting. I do whole body MRIs. I gained 10 pounds of muscle mass. And unfortunately, that didn't work either. It kept creeping up and was heading up and up and up. And finally, it got to, I think, about 6.8. No, I guess it was 7.0. And I said, all right, it's time for metformin. I took metformin. And once you know it, I'm not a responder. So the point out of all this is by the way, my genome predicted this. But the point out of all this is that I spiked my way in so as though something triggered it. The one question is, well, what works for me? So yeah, I was getting a little concerned when it got to 7.5 after still going on metformin, and it wasn't working. And the key was actually to do more experiments, of course. And what we discovered was that as we were following glucose disposition, that it turns out I'm insulin sensitive, and I also make insulin just fine. So what's wrong with me? Well, I actually don't dispense my glucose well, and it's because I don't release insulin well from the pancreas. And so I'm down here on one of these poor insulin dispensers, and because I just don't release my insulin from the pancreas. And so there's a Prandtl derivative, I never pronounce it right, rapinolide or something like that. You guys will know it better than me. And it turns out I'm a good responder to that. So I went from 7.5 to 6.3 right away. And that's keeping it in that range roughly. It sometimes creeps up. But so anyway, the point out of all this is what we call precision diabetes. By knowing what was wrong with me, we could prescribe the right drug to help get this under control. Also do walks quite a bit after I eat, and that even worked quite well. So anyway, the point out of all this is that we call, again, precision diabetes. By knowing what's wrong, you can make the right prescription and hopefully manage people's health better. Okay. One of the coolest things we learned from this study, and this is a big deal, is that no matter which home or other kinds of measurements we used, people's baseline turns out to be fairly stable. So it's a misnomer that we're changing all the time, blah, blah, blah. That's actually not the case. So no matter which home we use, metabolome, cytokine, transcriptome, this one's not as obvious, but if I showed just in 3D, it'd be obvious. Here's just 12 people with at least 10 visits. Each dot is a visit. Each color is a person. Okay, so I have 6 years of data here. I'm the light blue. What you'll notice is that people cluster, they're most similar to themselves, even over long periods, like 6 years. My dots all cluster together, and the red person's dots cluster together, and so on and so forth. So your omics profiles are very personal, they're very robust, and they distinguish you from the other person. So what that means, if you got up off your seat, we could tell who sat there by swipes, undoubtedly if we had the right reference. So the point out of all this is that your healthy profiles are stable. What's also quite interesting, I won't show you, but if you get a viral infection or do exercise or do what have you, you will shift your profile, but you will still look more like you than anyone else for the most part. There's some exceptions. So what that means, even when you get ill or something, you are still more like you. These personal profiles are very, very stable. And this is an important concept because this is where the GWAS guys say, oh, you need thousands of people, blah, 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 to tell differences between disease and not. And the answer is that's not true. The answer is all you need is your baseline. You can't miss a signature from a disease state and a healthy state if you're comparing yourself. But you'll miss it. You'll have a hard time if you're trying to compare the disease you with 1,000 other healthy people because you don't have the right reference. So this is why these healthy baselines are so important. We think it's really critical that you get your healthy baseline so you can detect disease at its earliest possible time because you're going to have a hard time doing it comparing you with everyone else. It's too late if you wait till you're diseased. And so you actually don't need very many people. In fact, you only need one to tell a strange you from a healthy you because you'll see that shift right away. All right. The other thing we've looked at, which hasn't really been explored before, is personal aging. So you probably appreciate that, people obviously change as they get old. And the way this has been studied historically is that people will basically, they'll look at old people and young people. It's cross-sectional. And they'll say, here are markers that are in old people that aren't in young people, like high hemoglobin A1C is, goes up. It's higher in older people than young folks. And so that's true. And so there's quite a few markers out there. But nobody's really studying aging in depth at a personal level. So what we did was we looked at our group because we're profiling them so deeply, we could actually see what molecules change over time. So I told you most don't, they're pretty stable, but there's 600 molecules that will gradually change over time. And we can start looking at them. And we find cross-sectionally all the same markers. Others find plus more because we're going deeper. But it turns out if you follow people individually, what you'll discover is that we all don't age the same. And I don't think this is a surprise, but nobody's looked at it. So for example, this is me on the left, a lot of markers here. I'm a pretty typical ager. My coagulation system goes up. So do some other metabolic markers and things you'll see in a minute. I'm a pretty typical person, except my immune system isn't aging so much, which is kind of interesting. Here's another person. He's a cardio-ager. His or her cardiac pathways are changing. And what's cool about this is you can measure these over a relatively short time frame. We can measure 5 measurements within 2 years is enough to tell what's going off. And we think this is a big deal because it's actionable information then. And as an example, you probably know there's a ton of supplements out there, Elysium, all this stuff that people do. They take this stuff at $60 a bottle. And for all you know, it's snake oil. You have no way of telling if it's working or not, because you have no way to tell if your personal aging is reversing or staying the same. So you're paying $60 a shot, they're making money and you have no idea if it's working. But if you have personal markers, you actually have a way of trying to follow this. Now, I do think this information is potentially actionable. What we did, I forgot. So we actually had 43 people with enough data that we could see how they were aging. And then we said, let's see if there are any major classes of aging that's occurring. And so from these 43 people, we could group them into 4 agetypes. Now there are more than 4 agetypes, like there's a cardio-ager, but we only had one of those. But if you look at in general and using statistical robustness, if you will, we can see what the major categories of ages are from this limited data set. And what we discovered is that there are some people are aging in all, so their kidney, liver, metabolic, and immunity. It's not just glucose, by the way, it's a lot of metabolic markers changing here. And these folks at the end, they're aging in all 4 categories. But there are other people who might age, for example, in 3 of the 4 categories, like this person here. I forgot to say, each column's a different person. So this person's aging in all, this person's aging 3 of the 4. I'm somewhere in here. I kind of lost track. I think I'm here, where I'm aging mostly in 3, but not so much in immune stuff. Here's a kidney ager, here's a metabolic ager, and a kidney ager, and so on and so forth. So you can actually see how people are aging at a personal level. And again, we think this is very actionable information because you could then act upon that. And so some of these pathways do have clinical markers because that's what the medical profession likes to see, although I think the pathway information is more robust, but nonetheless is what we see. And these are just 6 clinical markers we follow. This should say creatinine, missing another I and N. And in general, these markers behave the way they should, like hemoglobin. This last column is the cohort as a whole. It goes up. Same with creatinine, goes up for the cohort as own. So same with all these. Well, it's kind of cool if you look at individuals, you can see some are going up. A black line means it's significantly going up. And here's a black bar going down. This one means it's going down. So 4 people are actually increasing hemoglobin A1C, but 4 actually reduced it, meaning they've actually got it under control. And some of them have done it through diet, it looks like, some through, 1 through exercise, and 2 people through weight loss. So they actually, in this case, they actually took action and reversed this marker. Here is, this marker is going up, a kidney marker. There are, in fact, 10 people who are going down significantly. So again, the group as a whole, they go up, but these 10 people are going down. What's cool is that 8 of the 10 are on statins. And so it's possible it's related to muscle mass, but I'm not so sure because we don't see big changes in weight and stuff. So anyway, the point is that these folks have actually seemed to reducing and perhaps have improved kidney function. It'd be worth looking at further. I won't have time to get in as much because I want to get into some other things, but this just reinforces known clinical marker. It's called PhenoAge, set up by Morgan Levine's group. And basically, it just basically says what I told you. People are aging at different rates. Some people even, again, seem to be reversing their age, at least through these. This is a very limited marker panel. The point is that some people are, their biological age is above their chronological age, meaning they're older than they should be, and others are below, meaning they're younger. And some are aging fast, if that line's going up, and others seem to be, like say, slow or possibly even reversing. I, of course, turned out I'm smack on the line. It was very disappointing. I wanted to be below the line. And I, of course, was hoping that I was reversing my aging, but this is before I started weightlifting, so maybe weightlifting will had me reducing my age, we can see. All right, we've totally conflicted here, but as a way to scale this, everybody says, how are you going to scale this to thousands, if not millions of people? And the answer is form a company, and they're much more efficient, and it's much more medically focused. What I showed you was a research project, but just from the first bunch of people, the first little over 100, these are some of the early, there's more than this, but these are some of the early markers we've had, called some of the early pancreatic cancer, and that was found through longitudinal data. Some of the early prostate, ovarian, some of these were found because we have multiple lines of evidence that this is not right. And so again, we think these deep profiles can actually be scalable, can actually be applied to people who are healthy, and we hope will be actually powerful for better managing people's health. Now I want to tell you about some new studies we've just put out there that you might find interesting. This is a fun one run by Kevin Contrapas, Siwoo, and others in the lab with Francois Haddad. And this is to look at the effects of exercise. So you're told, right, exercise is great for you. Go do it, boost your immune system, better for metabolic control, all this sort of stuff. And nobody really knows how it works. And so the goal was to see if we can start getting some glimpses here. And for this, what we've done is some deep omics profiling following just a short bout of exercise. So here's the idea: 36 people, some of whom are insulin resistant, run to their VO2 max. So it's between 8 and 12 minutes. And I'm thinking, well, how much are people's molecular profiles going to change from a 8 to 10 minute run? And the answer is a lot. Turns out 9,000 of the 17,000 that we measured are all changing pretty dramatically. So we sampled them, I forgot to say, right after exercise, 2 minutes, 15 minutes, 30 minutes, an hour. We actually ran them out for 24 hours, but they eat here and it makes a mess after they eat. So anyway, we just look at the first hour, and what we find is tons and tons of things are changing, like I say, 9,000 things. And I'll just summarize a lot of data, but basically what we see is that some of the very earliest events are inflammation, oxidative stress, and some of the lipid signaling, which we think is a very early response. And again, it's engaging very important areas like oxidative stress is very important for aging and other aspects, obviously inflammation, important for your immune system and so on. And then some of the hormonal response, some of the metabolic response are a little bit later. There's a whole orchestra of metabolism that goes on. So instantly you get amino acid catabolism and some of the metabolic hormones kick in. But then you also, and then later, some of these branched chain amino acids, which are known to be important for metabolism. They're very interesting short chain fatty acids and medium and long chains, the way they're utilized during this process. And so the point out of all this is there's a very detailed choreography of your whole system, your whole biological system changes, even from a short bout of exercise. And we think that these things are very, very important for, again, your immune health, your oxidative stress, your metabolic health, and we can see the various fats that burn as well. One of the cool results we got is that, as you may know, VO2 max is a measure for fitness. And what we discovered is that we could have a 12 analyte panel that actually is pretty strongly predictive. It's over 0.8, and it's predictive value for VO2 max. So we think you don't have to run on a VO2, on a treadmill, take off all your clothes, run on this treadmill, spend a lot of money. We think you could actually get it right from a baseline blood draw in the future. That might be a good indicator of, in fact, your fitness. Now let's see how we're doing time-wise. All right, we're in pretty good shape. Oh yeah, one other thing we discovered is something we didn't quite get from the baseline measurements is that when people ran to the VO2 max, in general, a lot of things made sense, but there were 4 people very, very shifted in their profiles, either at baseline or after response to exercise. And it turns out one of those, some of them we haven't totally figured out, but one person turns out to be anemic. And looking back in hindsight, they were borderline. They were still in the healthy area for anemia at baseline, but they were definitely impaired upon exercise, and it turns out they're a thalassemia carrier. So maybe that's related to this. So the point is we could actually tease out defects from a stress echo, and people, that's why you do stress echoes, but we found something new that you wouldn't necessarily find the way people run this now. I want to say a little bit about pregnancy, since this is now everything I told you is about adults, and I think everything we're doing for adults we should be doing for kids, and I'd love to get that going. But this is a study run by Liang along with Mads Melby and others heavily involved in this as well. And so we decided, and David, Gary Shaw and David Peterson, Nima, and others are doing this as well via the Gates. But here's a study we ran with Mads, which is to try to understand metabolic changes at very, very high resolution through pregnancy at a level that hadn't been done before. So because we've been pushing this very hard, this is a big deal to us to see if we can metabolic profiling at a level no one's done. So what Mads had done, he'd taken 30 women, more now, but he sampled them every week all the way until birth, which is basically right there, the red triangle. So we follow these over time. We have postpartum samples. And then we do very, very deep profiling of metabolites from plasma. And we're following a ton of metabolites. It's really, again, quite deep. And what we discover, this part, not a surprise, we see, you know, here's where people are at baseline. They gradually shift their metabolites. And then after they have their child, they'll go back to baseline. So this is one way of looking at the data by combining all the features. And what we do is we dig in there and we see which molecules are changing. These are ones that go up over time. These ones are red, and these are ones that go down. These ones are blue. Some of them stay down after birth, but most come back. These pretty much always come back. And so you can see what's changing. Something's wrong here. This is steroid hormone biosynthesis. Some change amazingly dramatically, some are gradual. These are phospholipids, and certain other markers will go down and they'll come back. Again, some don't fully come back. And so we can see all the molecules that change. It's pretty cool. And then we can see the pathways that change. And it's going to jump to the punch here, but these are all the molecules and networks that are changing, which turns out to be pretty cool. But it's kind of fun that we can see when we look at molecules that change in general, we'll see those involved in obesity, which kind of makes sense with pregnancy. We need to compare that with some of the other work we've done in weight gain, weight loss. And also prepartum depression was kind of interesting to be the top pathway that as a disease pathway that showed up upon changing. So that is quite interesting that we have a molecular signature for this. We're not yet predictive. Now, one of the things we really wanted to do is see if we could predict gestational age and time to delivery. And so we took 21 subjects of the 30. We used that as a training or discovery cohort. And we found 42 molecules that worked pretty well. And they actually are quite predictive of gestational age. We tested on the other 9, and it actually held up. It's pretty good. 0.91 is a very, very strong predictive value. We then said, well, 42 is a lot. Can we find some others that would be, easier to manage? And so we came up with a 5 metabolite marker. 4 of them are progesterone derivatives, but not progesterone per se. And another's another new kind of molecule. And same thing, we ran it through here. And again, it captures most of the information, 0.85. And then on this validation's pretty good, 0.8. On this one, it's equally good. So 5 metabolites can predict gestational age pretty darn well, which we think is quite powerful. Not such a big deal in the US, but a very big deal in other parts of the world when people aren't quite as sure about what is, when that's happening, what is gestational age. And then these are the contributions of the different 5 markers. So we've gone on and actually taken 3 of the compounds. We start classifying women about when they would be, where they're at and where they would be relative to term date, say 34. So we can set up markers that, again, predict where you are relative to preterm versus post-term. I won't have time to get into this in a lot of detail, but then we can also classify women as the probability that they may have, whether before these different time periods and when they would actually time to deliver. And I might just jump ahead. This is easiest to probably explain here. So the point is we can actually classify, if we take some markers from 3rd trimester, we can classify time to delivery with reasonable accuracy. It's not perfect. This is just one of the markers that work. This one marker works great on, these are individual women. It works great on all these, not so good on this. Is why we have 3 markers to make better predictions. And same thing for 2 or 8 weeks time to delivery. We can make some prediction about whether they're likely to deliver within the next 4 weeks or 8 weeks from sampling in the 3rd trimester. So we think this could have value actually, and I should say I'm totally conflicted because we're involved with the company that's going to try and commercialize this. All right, so in the end, we hope that we can actually ultimately set up a nice time to delivery date, a gestational age date as well. And then maybe these are very interesting molecules for other reasons as well. All right, I want to say if we have time, I think, can I take 5 more minutes there, Tony? I'll take that as a yes, since I don't, yeah, we're probably muted there. All right. Anyway, we're very keen on wearables, and I think this is a big deal for not only adults, but for kids, and especially for what I'm about to say regarding infectious disease. So some of you know we've gotten into this in a big way. And we started this about, I think it was 7.5 years ago before the Apple Watch was out. People were getting these smartwatches as fitness trackers, and they figure out their fitness, throw them in a drawer, be done with them. And we realized that these are actually pretty powerful physiological monitors. And we realized that maybe we can actually use these to continuously measure people a little bit better than the every 3-month sampling we're doing while people are healthy. And they're powerful because they make hundreds of thousands of measurements daily. They're now capable of measuring many, many different things. And now they're also worn by millions of people. So we actually have a far reach when we start developing technologies around these. So what we did was, the way we use these, I'm just going to tell you about the Smartwatch data. We were doing a lot with glucose monitoring and environmental exposures, both of which are very, very relevant to kids. And so that's another whole wearable sort of talk. It's pretty interesting. But I want to go around the infectious disease because I do think it's relevant to everyone, kids and adults, probably more for the aged. But anyway, what we do with these is we actually take a smartwatch and we start using it to tease out people's circadian patterns. So sleep, heart rate, skin temperature for this particular watch we're using at time. And so you tease out individual patterns. People, this is my circadian pattern: sleep at night, wake during day, occasional nap, sleep at night. Heart rate low at night, higher during day. Skin temperature, which is different from oral temperature, is high at night, drops in day, comes back for most people, not all. All right. So what we discovered is that one thing's pretty clear already. Sorry about that. So one thing that's pretty clear already is that we could start telling deviations from baseline. We discovered that people's blood oxygen drops on airplanes. That's known already. It's not well documented how far it drops. It drops pretty far, actually, for most people. And then, but what we also discovered is that we could start detecting infectious disease. And in fact, we discovered my Lyme disease with a simple pulse ox and smartwatch. And this is, the backstory was I was helping my brother put up fences in rural Massachusetts. And I was flying to Norway through Frankfurt. And on that last flight from Frankfurt to Oslo, my blood oxygen dropped abnormally low. This is what it normally drops on a short flight like that. This one dropped to median value of 90 instead of 96, and it never came back to normal after landing. And then I also noticed time my heart rate was high, and I later learned my skin temperature was up. So the timing was such, I had been helping my brother put up fences in rural Massachusetts 2 weeks earlier. So I thought it might be Lyme disease because of the timing. And I went to a doctor in Norway. I got a low-grade fever after seeing this initially. And I kind of went to him and I told him it might be Lyme, and he drew blood, and sure enough, my monocytes are up. Said, yep, you're sick. Sorry, just accidentally lost my screen. Here we go. And he basically said, all right, you got an infection. He recommended penicillin. I said, no, I think I need doxycycline. A little tense for a few moments. He gave in and gave me doxycycline. And then you take it for 2 weeks, which I did, cleared up right away, the fever and all that stuff. And then I measured myself when I got back, and sure enough, I was Lyme positive by sera and antigen test. And what was nice, I had a very well-controlled experiment. I'd given blood about 3 days before I left, and I was negative. So I sera converted during that time. So what was powerful about this is that from a simple pulse ox and smartwatch, we could tell when I got ill from Lyme disease. So that prompted us to write an algorithm, prompted me to look at 2 years of data I had at the time, and I'd been sick 4 times. This is a big deal. Once was Lyme, 2 times are viral, this one and this one. And this time I was asymptomatic, the circle time, yet it turns out I had high CRP, meaning I was ill. It's just as high as these other times. So the point out of all this is that I had elevated CRP, was ill 4 times, one of which I didn't know. I was asymptomatic. And which, every single time was high percent heart rate or outlier heart rate. You can get it from median too. This is skin temperature. Every single time I was ill, I had elevated skin temperature or heart rate. So we wrote an algorithm called change of heart. It doesn't work so well for skin temperature. I think there are technical reasons for that. It's also true that skin temperature is not the best measurement for getting ill, we think. I know where everybody's told stick a thermometer in your mouth when you get ill, but I actually think heart rate's a better measure than skin temperature. I don't know why we're so focused on skin temperature. Anyway, this is like a change from baseline. Again, know your baseline, find the change, the sliding window. Every single time I was ill, I could pick it up as this change in heart rate in advance of either certainly before I gave blood, and either at or before I was symptomatic, every single time. So we think your heart rate goes up before you're actually feeling symptomatic. And we had 3 other people, one of whom was sick twice, same thing, that algorithm works every single time on those. So naturally, with this latest pandemic, we said, well, can we apply this to COVID-19? So we launched a study, very interesting, trying to launch a wearable study with IRBs these days, but they did approve it. And we were partnering with Fitbit, Garmin, others, every company. We're device agnostic. We've enrolled a ton of people. You guys can enroll too. And we've mostly concentrated on COVID-19. This is now about 50 people who are COVID-19 positive, who know their symptom and diagnosis state. That's a big deal. And so what we're doing here, we're starting with this group, 2-phase study. We want to train algorithms to optimally detect when you're ill with COVID-19, and then we're going to ping you and tell you when we think you're getting ill. It may not be COVID, it may be anything, but that's the 2nd phase, which we have not rolled out. So it turns out, oops, I got the wrong slide. This one has dates, but anyway, permission from this folk. So the point out of all this is that this is the very first patient we had. This purple is when they were actually diagnosed. This red is actually when they were symptomatic. And what you'll notice, their change from resting heart rate jumped up on the 9th, 10 days early. It's 9.5 days early, it turns out, before they had symptoms. So this person was spreading the virus for 9.5 days without knowing they had the illness. And we've set up a way to do this online. This is tricky because what we want to do, this is retrospective. Just say, oh, when is your heart rate up? We can find that. But the goal is to find out when you're up relative to your healthy baseline. So you build a normalized distribution of what your signals normally look like, and then you see when you exceed that in a statistically meaningful fashion. And sure enough, we can set this up. So we call this online detection. And this is what we're going to roll out in the 2nd phase to tell you when your heart rate signal is up relative to what it should be in it. A 6-hour window by far is enough. You can actually get by with less. And so again, we think this will be very powerful. We have other people enrolled in the study. And so the other thing that's really, really important is that we've set up a dashboard and a cloud computing system. This is a big deal. This is a scalable format that's highly secure that can actually work with millions of people. So suck in their data, display it in a usable fashion. So you can look at whatever data you want at whatever resolution you want. Because I think the person is going to be the ultimate gatekeeper of information. It will augment what a physician can offer, but people have more time to spend on themselves than a physician. And you'll be able to share this with your physician so they can actually see your heart rate and all these changes. And then back on this study, so we're going to ping you when we think you're getting ill. Get ready. I'm hoping to roll that out soon. We just submitted that to the IRB. And so in the long run, this is Mike Snyder's future. Again, I said at the beginning, I see us getting people's genome sequence before they're born. With much deeper molecular and wearable profiles on people, we can much, much better predict disease risk, catch disease early. And I'd like to see a lot of what we're doing get applied to kids. So I've had an amazing team. The most recent group leading this has been Wenyu Xiao, Brian Pining, and others, Kevin Contra. We're collaborating on the microbiome with George Weinstock, Tracy McLaughlin, great clinical collaborator. Come to this in a bit. Let's see, the Wearables was started by Xiao, Jesse, and others. The pregnancy work was really spearheaded by Liang Liang, worked with Mads Melby. And then we've just assembled over the last 2 months an incredible wearables team. It's pretty intense to actually go at this. We're going to try and we want to have impact in now, not in the fall if we can. So we're trying to really get this working well. And I forgot to say, but it works in about 3/4 of cases. We can tell either a signal at or before time of diagnosis, and most of that's at or before the time of symptoms. And so we can see these strong signals, again, either before when people are symptomatic, most of the time. We are missing a few. It's not working perfect. And I think it's not working perfect. Turns out we know in some cases because they have actually respiratory issues going on. And so it's not quite working in some cases. And we're still trying to understand in all cases, it should work every time. Why isn't it working? That's what we're trying to figure out. So that's the story. Happy to take questions if you have any. And I hope that was pretty clear. I know it's very adult focused, but again, you can see all these technologies should be powerful for kids as well.
Wow, thanks so much, Mike. That was quite a tour de force, both for the adult as well as for the maternal and child health. We have quite a few questions, way more than we can actually have time to answer. So I'm going to try to group them into different areas that are all about the same idea. The first one is about if you don't get anything from this talk, it's about you need a personalized baseline to what we're hearing. And so the question is, how many measurements and what sort of measurements would you, over what time frame for a healthy person would you recommend to start off?
That's a good question. You know, even one's better than nothing, but 2 is pretty good because you'll see you're fairly stable, and that'll you'll get pretty reasonable measurements if you measure yourself twice. In our ideal world, you do 3. So what QBio is saying is try and get 2 early because it really will pin down your baseline. And we believe, you know, we're sampling people a lot, just naturally as part of our study. But I'll tell you, even 1 is powerful because there was someone who came up to us. We kind of been bugging them. They were predisposed for a disease, and we actually figured out what it was from the genetics. They kept saying, you got to get a profile so we could understand better what's going on. And they said, yeah, we'll do it. And then finally, they only came to us after they got ill. Their son got ill as a kid. Right. And I was saying, gosh, I really wish they'd come in earlier because we would have had that comparison.
And over a time frame, I mean, if you had, would it be over 2, like if you had 2 or 3 measurements over a year, over 6 months?
Oh, yeah, we like to take 2 right away in a couple of months if we could, say 3 months, but even 6 months would be good, or certainly 3 within 6, that would be ideal. We honestly don't know the answer to this, though, and this is one of the things we want to discover. How often should you sample people? Right. And nobody really knows, and I think we can build personal models around that, and maybe you don't have to do such a deep profile every single time once you see what your trajectories are doing, if you know what I mean. So we're going to build those personal models. We already are with the wearables, by the way.
Yeah, that's awesome. And so the next group of questions is about, on a practical level, you know, screening tests for low probability events are very expensive. But if you have a lot of measurements that are predictive, where you're measuring you against yourself, that's a whole different, you know, sensitivity and specificity argument. So given the cost, given sort of the cost coming down of the measurements, and how long do you see, or how long in the future do you see where actually it becomes cost effective for insurance companies to basically force or recommend people to get these measurements?
Yeah, that's a great question. So the answer is QBio charges $3,500. They'll give you a whole body MRI and pretty deep profile. It's not as deep as what we do in our research study, but it's much more medically oriented. So physicians like you, Tony, would know what to do with that information. So it's a much more actionable set of information. So you can argue, is $3,500 too much or too little? Compared to what happens when somebody gets a disease late, it's a good deal. You know, but it still could be high. And the biggest problem, I mean, first of all, I know we can drive those costs down as we do more multi-animal assays. As we start driving things towards measure 500, several thousand things at once, that cost of that test is roughly the same as measuring something once.
Right.
So I think we can drive costs way down to a few $100s, although it tends not to be what happens. Usually add on more molecules. Tests really go down. So anyway, I think we can drive it down if we had to. And I still think at some level that's a good deal, but I think we have to change the whole healthcare system because nobody pays to keep you healthy. That's the problem. It's a payment thing.
And then so then your proposal then would be to actually integrate this information into like an Epic or a patient health record over time that's available. Right? That's a sort of idea.
Totally. Yep. So absolutely right. So you take all the medically actionable. Nobody knows what to do with an RNA-seq experiment,
right?
But we'll know what to do with some of the key markers that are associated there. Yep.
Yeah. So there's a lot of questions about precision aging. That sort of rang the meter from our group. The question is about how do those measurements relate to like telomere length, neurodegeneration, as well as neurological health? Because a lot of data on neurological aging, which you didn't talk about.
Yeah.
But, is it brain derived, or is it actually when you talk about metabolic disease? Because a lot of biological data about how the brain aging actually controls the body, but you're telling me that the body controls the rest of it. So how do you put?
Well, I think they go hand in hand. So there's systemic aging where, and so I look at aging like a car. That your car as a whole gets older and older and things, but some parts wear out faster than others. And in some people, it's, neurological issues like Alzheimer's and things, and especially if you're genetically predisposed, just like your car, your engine could wear out faster than the chassis or something. So there's different things will wear out at different times, and that's how I see that. We do pick up some neurological markers. I just didn't have enough to get statistical significance from the group we're looking at now. I think as we add on more people, we'll pick that up. We do see some markers, by the way, changing, but not enough to make any statistically significant statement. It's a great question.
And then we have a whole group of questions about other omes. So one very insightful person asked about the sociome and other environmental, and those, how do you, and that's especially for maternal and child health, where childhood development is clearly environmentally regulated or influenced for like chronic depression and childhood depression, that sort of. How do you see us measuring the other types of omes that are more environmental?
So I didn't talk about that, but some of you may have seen me talk about this before. We have an exposometer, and I've left that out simply because it wasn't time. And we have a funded study around Crohn's, believe it or not, although we're going broad on that. So this thing sucks up all the particulates in the air, captures all the particulates, but also underneath has a chemical absorbent. So we can measure all the chemicals and biologicals you're breathing right now. So if you have Crohn's, join our study, or you have a kid with Crohn's or something like that. And so we think this is going to be powerful because right now, I'm sure everyone listening has no idea what they're exposed to for pollen and things like that. And we think this is a huge deal. And it's unfortunately going to be a very long-term study to know how that correlates with your health. But we're already learning some things. I know what I'm allergic to now that I didn't realize before, thanks to measuring this stuff. Pretty simple. And chemicals, by the way, you'll see DEET, certain pesticides, certain carcinogens, they're everywhere, which is also very eye-opening.
Right. I think that's going to be really amazing. And then I think the last couple of questions are around maternal and child health and pregnancy. So
Okay.
Can you talk about the predictive, the study that's coming out where it's predictive value? I know you have already measurements of, with Steve Quake has measurements of what you're telling about pregnancy, but how effective in terms of early disease detection, for example, folic acid in maternal diet. I mean, how much can we tell how sensitive do you think those measurements are and how useful?
It's a great question. So we're measuring those now. Liang is measuring vitamin D and all these things in women. We haven't targeted so much for that around nutritional markers. It's probably in our data. We just haven't looked at it. We mostly grab known things. So I know we can pick up folic acid and should look at that. I don't remember if we saw what we saw on the folic acid. But anyway, that is a big deal, we think. And I forgot to mention Steve. Steve's cell-free work is really fantastic. We actually get better predictive value from metabolites. And ultimately, you know, I'd love to see this as a home test where you just dip in. And my own view, by the way, of all this is that a lot of it's going to be a home test, not MRI, whole body MRI. But there's no reason, if you think of everything you do now, it's like shopping and stuff. My family, I'm the only guy who goes to stores, and I don't now because of COVID. But you know, they order everything online, and I see no reason medicine isn't done totally online as much as possible. Why do you have to go to a doctor to get blood? You could give it and mail it in. So
Right.
I'm a big believer we will become Amazonized in medicine.
Right. And then the last question is, one of the issues is collecting data on the fetus, non-invasively, so that understand how. Do you think that getting blood from the mother is enough to then deconvolute what's happening with the fetus?
That's a good question. I hope so. I honestly don't know how far we can push it, right? You're probably looking at mixed health when you're measuring the mom, unfortunately. And so we'll just have to see. And some markers are suggested to be, we see placental markers, by the way, in the
Right.
mom. So that part's pretty clear. But how much is specific for the fetus versus the mom, not sure.
Think you'd be able to deconvolute if the mom's like this and then the baby's like that, then you can deconvolute the pattern?
I hope so. And this is where multiple measurements is a big deal, right? If you can make thousands of measurements and you know certain things are always correlated in the mom and correlate differently in the fetus, you can deconvolute that. You should be able to.
Great. Well, we've run out of time, but thank you so much for a phenomenal presentation and also for your amazing contributions to maternal and child health. I mean, really, I think the measurements and the baselines we're getting will be just changing and transforming the way we do medicine. So thanks, Mike.
Thank you.
So I'll turn it back to Roxana, and but thanks everyone for joining us. We'll see you again September 11th for our next MCHRI symposium. Thanks very much.